Thank you. Thank you so much to the organizers for the invitation to present and to participate in this important discussion. I am an assistant professor at the University of Arizona College of Medicine, and my research focuses on the impacts of the lighting environment on sleep and circadian physiology in childhood, with a particular focus on preschool age children. And as you might imagine, there are a lot of methodological challenges in conducting controlled circadian protocols with this age group. So today I'm going to talk about what we have managed to learn regarding the circadian responses to light in this age group. Some of this is older data, some of this is newer data, and then I'll also touch on the many open questions that still remain. Here we go. So first off, we know that light exposure is crucial for circadian regulation even in infancy. Findings from several studies indicate that exposing premature infants in the NICU to a light-dark cycle rather than constant dim light conditions is associated with earlier emergence of day-night differences in activity, so an earlier rhythm, faster weight gain, reductions in fussing and crying, and shorter hospital stays. Additionally, in a study of full-term infants from 6 to 12 weeks of age, those who were regarded as sleeping well at night were exposed to significantly more light in the early afternoon, emphasizing the importance of a healthy light diet for early sleep and circadian development. But jumping ahead from infancy to the preschool years, and these unpublished data from healthy children ages 3 to 6, we find that those who spend more time each day exposed to bright light of greater than 1,000 lux have significantly earlier bedtime, sleep onset times, and sleep offset times. Earlier sleep timing was also associated with greater average light levels across the full 24 hours, as well as when restricted to just daylight hours between 7 a.m. to 7 p.m., highlighting the consistency of this relationship between daytime light exposure and sleep throughout early life. Interestingly, we did find that this relationship was still present in school-age children aged 8 to 9, but not in adolescents aged 15 to 16. And so whether this is related to the known decrease in circadian sensitivity to light in adolescence, or just to changes in adolescents having more autonomy over their activities and their sleep schedules, would still be an open question for this population. But we know that light exposure also plays an important role in the timing of the circadian clock in early childhood. In this study, 21 preschoolers followed a stable sleep schedule and wore a pendant light meter for 4 days, and then completed a dim light melatonin onset assessment. And when controlling for their bedtimes, the children who had been exposed to more evening light had later melatonin onsets. And in a hierarchical linear regression model, evening light exposure accounted for about 13% of the variance in DILMO timing. In a subsequent study examining the early melatonin suppression response, children were exposed to a bright light stimulus of 1,000 lux in the hour before their habitual bedtimes. And during the light exposure, children exhibited very robust melatonin suppression with an average of about 88% compared to the melatonin levels on the previous evening collected under dim light conditions. The melatonin levels also remained attenuated even 50 minutes after the end of the light exposure, indicating a very sustained response. So in adults, we know that light exposure in the evening both suppresses melatonin and delays circadian timing in a dose-dependent manner with intensity. And much of my postdoc work centered around developing illuminance response curves to evening light in young children. And as we just heard, children are much more sensitive to light exposure than adults in terms of their circadian response. And so we did expect to see these curves would be shifted a little bit to the left, but we did predict that we would see a similar dose-response relationship. A little bit about the way that we measure these things in young children. We conducted an assessment of their baseline DILMO under dim light, and then children were exposed to light for 1 hour before their habitual bedtimes, which ranged in intensity from 5 to 5,000 lux. And as you can see in the picture here, the way that we get kids to do these types of protocols is they play at a dimmable LED table in order to make sure that they're looking down continuously for a full hour to get that light exposure. Young children are also unable to provide saliva samples via passive drool. And so the way that we collect saliva for melatonin is have them chew on one end of a braided cotton roll for about 2 minutes, as seen in this picture, which is then placed into a tube and centrifuged. Lastly, this protocol required children to be under constant dim light conditions for 3 days. And as you can imagine, that would be a challenge to get preschoolers and their parents to live in a sleep lab for 3 whole days. And so we conducted all of these protocols in participants' homes where we recreated well-controlled laboratory conditions. And so I always like to show an example of what that looks like. Here's our team in the process of converting a home into a dim light environment. The first thing we have to do is cover every single window in their house with black tarp. And then we ensure that all light levels in the home are less than 10 lux at the child's angle of gaze, which involves adjusting or disabling all home light fixtures, in addition to identifying any appliances or toys or children's toothbrushes and shoes, anything that lights up at all. So that really highlighting the challenges and the need for getting creative when you're translating circadian protocols into something that's feasible for young children and their families. But we did manage to complete this initial protocol with 33 healthy, good sleeping children ages 3 and 4 years. And instead of those dose-dependent illuminance response curves that we see in adults, this is what we observed in terms of melatonin suppression during the 1 hour light exposure across the different intensities. You can see that children had a very high level of melatonin suppression in response to light across the full range of intensities, with an average suppression of 86%, and no clear relationship with light intensity was observed. And then here's what we see in terms of circadian phase delay. Across all of the intensities, we observed an average phase delay of 56 minutes. And again, we didn't observe any clear relationship between light intensity and the magnitude of the phase delay. Rather, we see very robust phase delays across the full range of intensities, even in response to the lowest intensities. And we know that circadian misalignment in early childhood is associated with behavioral sleep problems, in part because young children do not generally get to choose their own bedtimes, and they also have a much narrower phase angle of entrainment with bedtime than adults typically do. And so as parents select that bedtime gets closer to children's melatonin onset, or if parents are trying to put them to bed before melatonin onset, they take longer to fall asleep and have increased bedtime resistance, likely because they're being asked to go to bed at a time when they are not physiologically ready to fall asleep. So if children's domos are being pushed later by evening artificial light exposure in the magnitudes that we're observing in our data here, a mismatch between their parent-selected bedtime and the circadian timing could be contributing to the high prevalence of sleep problems that we observe at this age group. We did also observe very large individual differences in the magnitude of the circadian response among the children. So in this figure, we have individual melatonin curves for 6 of the children, with the baseline night presented as the orange line and the final night after the light exposure in blue. And the top 2 panels depict 2 children who received low intensities of light at a close circadian time, the middle panel a medium intensity of light, and the bottom panel a high intensity of light. And within each pair, despite the similarities in the circadian timing and the intensity of the light exposure they received, we observed large differences between the children, with the phase delay in each pair differing by 30 to 40 minutes. And this is consistent with work in adults showing large individual differences in circadian sensitivity in response to an evening light exposure. So in this study, they found that 50% of the maximal melatonin suppression response occurred in one participant at only 6 lux, and not until 350 lux in another. And there's a variety of factors that have been hypothesized to underlie individual differences in photosensitivity, much of which is going to be touched upon today and tomorrow as part of the symposium. So that includes age, sex, chronotype, genetics, pupil size, and even eye color, though how these different factors might interact to produce individual differences in young children is still yet to be explored. We next examined circadian sensitivity to evening light spectra instead of intensity in preschool age children in a within subject study. And you can see the 2 light conditions that we used for this study depicted on the right, which we held constant at a photopic illuminance of 20 lux. So given how sensitive the young children were in our previous study, we wanted to make sure we weren't fully saturating the response. The 5,000 Kelvin light source on the bottom is very similar in spectrum to the light in the first study. So we expected that the participants would have a high sensitivity to that condition, but we wanted to know if compared to that, exposure to a warmer light containing less blue light would result in less melatonin suppression in these young children. And in the figure on the left, you can see that there was indeed significantly greater melatonin suppression during the 5,000 Kelvin higher CCT light exposure compared with the 2,700 Kelvin condition. Melatonin levels also remained significantly lower 20 minutes after the end of the light exposure for the 5,000 Kelvin condition compared to the baseline night under dim conditions, but not after the 2,700 Kelvin exposure, indicating there was a more sustained response to that higher CCT light as well. So altogether, the findings from these initial studies provided evidence that the early circadian system is highly sensitive to light exposure in the hour before bedtime. But it's obvious that there's still a lot that we don't know about the circadian response to light in early childhood. Although the data that I presented on melatonin suppression and phase shift provide a crucial snapshot of the response to light in this age group before bedtime, our knowledge about the full phase shifting effects of light in this age group still remain pretty limited. We did collect data that remain unpublished to examine the dose response relationship between morning light and the phase advancing response in preschoolers. So here we gave a 1 hour light exposure starting at children's habitual wake time, with light intensities ranging from very dim at 1.5 lux as kind of a control condition up to 2000 lux. However, this was a really tough protocol to recruit for because we woke children up an hour and a half early and kept them up an hour late for 3 consecutive days. And so we ended up completing only 11 participants by the time the grant ended. And so you can see here, we really didn't see a consistent phase advance, with phase delays being below the line and phase advances being above the line. In fact, we often saw a phase delay even in response to the brighter intensities of light at 200 and 500 lux. We were only able to calculate phase shift for one participant at the brightest intensity of 2000, and they did show a phase advance, but it was only for 12 minutes. And so it's obviously hard to draw any significant conclusions from such a limited data set. But one possibility is that it just takes a much brighter morning light to elicit a consistent phase advance in this age group. Although that would be surprising given how sensitive they seem to be even to low levels of light exposure in the evening. It's also possible that our light exposure was given at the wrong circadian time to elicit a strong phase advance response, which we can't know at this point without a phase response curve. However, establishing a true phase response curve to light runs against the obvious methodological limitations of trying to run a forced asynchrony or ultradian protocol with young children. The only phase response curve to light with any pediatric population to date that I'm aware of was conducted with adolescents aged 14 to 17. And the maximum period of phase delay corresponded with habitual bedtime, whereas the maximum period of advance occurred shortly before wake, suggesting that we probably were within the right zone, assuming that something would be similar in young children. Surprisingly, the authors noted that phase advances also occurred in response to light in the afternoon. However, subsequent follow-up studies have failed to replicate this finding. But given the known changes in circadian photosensitivity between childhood and adolescence, knowing how the circadian effects of light in early childhood change across the cycle is crucial in order to identify and test feasible light-based interventions that can be accomplished with this age group and that are effective in improving circadian health. And our next work is attempting to begin to examine this. I've recently received funding to conduct a study on whether we can actually reduce evening light exposure in children that have sleep onset difficulties in order to advance their sleep and circadian timing and improve sleep health outcomes. So we're examining both the feasibility, can we even do these kinds of interventions with young children, as well as preliminary efficacy of either changing the lights in the child's house to smart bulbs that will automatically get dimmer and redder in the hour before their bedtime, as a passive intervention that really doesn't require much from the child and their parents, compared with having children wear blue light blocking glasses in the evening as an active intervention compared with clear placebo glasses. So we are just about to start data collection for this study, so stay tuned for hopefully some interesting data in the next few years on that. Additionally, in thinking about ways to improve early circadian health, there are data in adults showing that insufficient light throughout the day increases the impact of light at night on the circadian system, suggesting that sufficient daytime light exposure kind of inoculates the system against the disruptive effects of light at night. And so in the circadian protocols of preschoolers that I showed, the experimental light exposure occurred after the children were in dim light of less than 10 lux for 27 hours. So it did likely somewhat increase their sensitivity to the circadian disrupting effects of the evening light exposure. But to my knowledge, no research to date has examined whether bright light throughout the day would be similarly beneficial to protect against the effects of light at night in young children, who we see have this really high photosensitivity to those light exposures. But such data could support recommendations related to better lighting in schools, greater outdoor playtime to improve early circadian and sleep health. And so we're currently working on obtaining funding in order to examine this in this age group as well. There are data from one study in children ages 5 to 15 that shows that greater light exposure in the prior 24 hours is associated with a reduced melanopsin-driven pupil response to a 1 second light exposure delivered in the morning. So it suggests that there would be hopefully some beneficial effect of daytime light exposure to reduce circadian photosensitivity in early childhood. But how this would translate to that circadian response to light at night remains to be examined. Lastly, recommendations from a recent expert-led consensus panel on light exposure for healthy adults suggest that light with a melanopic EDI of over 10 lux should be avoided at least 3 hours before bedtime. However, these recommendations likely need to be adjusted for children whose parent-selected bedtimes often occur near or before sunset at certain times of year. In fact, the average bedtime for our healthy, good sleeping preschoolers was 8:03 p.m. during the summer in Boulder, Colorado, where sunset can be as late as 8:30 p.m. And we see that in the 3 hours before their bedtimes, they were exposed to an average light of 434 lux, so well above recommended levels for adults during that time. School-age children had reduced, but were still exposed to over 100 lux on average in the 3 hours before their bedtimes, with that value finally dropping to just 30 lux for adolescents. And so increasing our understanding of normative patterns of light exposure and how light affects the maturing circadian system, are crucial for developing age-appropriate recommendations for healthy light exposure across childhood. With that, thank you all so much for your attention, and I greatly look forward to the discussion during the upcoming Q&A session. Thank you.